Hello, this is Matt Dean with A Plus College Ready, and today we're going to talk a little bit about biotechnology. First of all, we'll start by talking about genetic engineering. Sometimes this is also called genetic modification. This is a process used to alter the DNA of an organism's genome. It can be something fairly simple like changing one base pair, deleting a whole region or part of the DNA, or putting in a new copy of a gene, maybe from even a different species of organism. This can also be a process in which DNA is extracted from another organism's genome and combined with the DNA of the individual. Genetic engineering is used to enhance or modify characteristics of an organism. It can be used with any organism, all the way from a bacterium, all the way up to maybe even a human. It can be used to produce plants that have higher nutritional value, livestock that grow bigger. It can also cause plants to tolerate exposure to certain herbicides. Biotechnology is the use of living systems, organisms, or parts of organisms to manipulate natural processes to develop products, systems, or even environments that will benefit people. Biotechnologies often or can be used to produce pharmaceuticals, to produce food. It can be used in waste management, water purification, and you'll also sometimes hear it called genetic or biomedical engineering. Another important term that we need to know about is recombinant DNA. This is a process of taking DNA from two different organisms, usually two different species, and joining them together to make a new piece of DNA. This new piece of recombinant DNA is then inserted into a host organism, oftentimes it's a bacteria, to make a new gene product. This is how, for example, human insulin is made in bacteria. Some possible applications of biotech and recombinant DNA are, first of all, gene therapy. Even though this process is right now fairly experimental, we're hoping that one day we can use genes to treat and maybe prevent disease. Instead of using a medicine, we could insert a gene and do away with the need for the drugs or surgery. Biotechnology is currently used to produce lots of pharmaceuticals. I've already mentioned human insulin. It's made in bacteria. Human growth hormone. Also, some vaccines are produced by biotech. We all know that biotech is used in the process of creating DNA fingerprints, which are used to identify criminal suspects. It can also be used to perform very accurate paternity or maternity testing. And probably at this point, the most important application of biotechnology is in agriculture. Biotech has been used to create all kinds of genetically modified foods or GMOs, genetically modified organisms. Many crop plants have been modified to become herbicide resistant, Roundup resistant. And this allows farmers not to have to till. Instead, they can spray their field with Roundup, kill the weeds, plant their crop plant. The Roundup doesn't kill the crop plant, so it grows without having to till the ground. This saves lots of money for the farmer. Other crop plants have been modified to create larger and hardier fruits, and even others have been modified to become cold tolerant or resistant to certain diseases. So let's talk a little bit about the cloning of DNA. This is the process of making multiple identical copies of a particular piece of DNA. Usually it starts off when the gene is inserted into a little circular piece of bacterial DNA called a plasmid. This insertion is usually performed using enzymes called restriction enzymes. These enzymes cut the DNA, and then we can use another enzyme called ligase to essentially paste it in and make the connection permanent. This produces a molecule of recombinant DNA. It's part bacterial and part whatever organism we're using to insert the gene from. So this piece of recombinant DNA can then be used to make copies in this process of DNA cloning. Those copies that can then be used to make a lot of protein, for example, the way insulin works. They might be analyzed through some type of test. But anyway, moving along. After we make the recombinant DNA, To copy it, that recombinant plasmid then has to be introduced into the bacteria. This process is called a transformation. Bacteria that are carrying the plasmid are then selected for, and they're grown. And every time they reproduce, they replicate that plasmid. That's why this is called DNA cloning. And they pass it on to their offspring. So let's get a little bit more details about the steps in the process. First of all, a bacteria plasmid is cut with restriction enzymes. These enzymes were isolated from bacteria, and they were actually used by the bacteria to cut up the DNA from bacteriophages, viruses that infect and can kill bacteria. They cut the DNA after very specific nucleotide sequences of A's, T's, G's, and C's. And the places that they cut at are called restriction sites. You'll see that restriction enzymes often have really weird names like EcoRI or HIND3. They're often named after the bacteria that were discovered in. For example, EcoR1 was discovered in E. coli. Many restriction enzymes cut the DNA strands of the DNA molecule at different points, and thus leave behind unpaired sequences called sticky ends. For example, as you can see down here in my picture at the bottom, this particular enzyme cuts along this green line, so that once it's finished, this top piece is going to have an AATT sequence that's going to be unpaired, and the bottom part is going to have a TTAA sequence that's going to be unpaired. Those are called sticky ends because if we put a complementary strand of DNA nearby and they will attach to each other with hydrogen bonds and form a connection. Once those pieces bind with the hydrogen bonds, the enzyme ligase is used to permanently connect the strands together. And at this point, we have a piece of recombinant DNA, a recombinant DNA plasmid. So here we can see the process. So here's the restriction site of the DNA that we want to cut. The restriction enzymes cut the DNA, leaving behind the sticky ends that we can see in the picture. The gray fragment is the piece of DNA we want to insert into the plasmid. Notice that its sticky ends are complementary to the sticky ends of the plasmid, the bacterial plasmid, the green piece. And then once they stick together, form those hydrogen bonds, ligase is used to seal those strands and make a permanent connection. So let's look a little bit deeper at the steps in the process. Again, first of all, the plasmid is inserted into the bacteria. Once that plasmid is in there, the bacteria are said to be transgenic because they have DNA from another species inside them. Oftentimes, the plasmid also contains a gene that codes for resistance to a particular antibiotic. To get the plasmid into the bacteria, we use a combination of chemical treatment that makes the cell membrane permeable to the plasmid and also heat shock, and that allows the bacteria to take in those plasmids. Even though we're using these chemical treatments and heat shocks, most of the bacteria don't get transformed. The process is pretty inefficient. So all the bacteria are then cultured in a medium, agar type medium, but the medium also contains an antibiotic, the one that the plasmid codes for resistance to. Because that antibiotic is in the medium, the bacteria that did not get transformed, they're killed by it. And only the bacteria that are transformed live, and they're artificially selected for. Those bacteria, the selected for ones, are then cultured usually in large vats. Again, every time they reproduce the plasmid, and the bacteria essentially become factories that create the protein coded for by the inserted gene. Those proteins can then be harvested and purified. And like I said, this is where we get human insulin from bacteria, also human growth hormone. So again, we can see the basic process in this diagram. So the gene is isolated from the source. It's inserted into a bacterial plasmid. The recombinant plasmid is then inserted into the bacterial host using a combination of chemical treatment and heat shock. The transformed bacteria are selected for, usually using antibiotics. Every time the bacteria reproduce, they reproduce the plasmid. And then that plasmid can be used to create proteins like insulin, growth hormone, proteins that help to dissolve clots in heart attack therapy. They can be used to do all kinds of things. So that is DNA cloning. Another process that we want to take just a minute to talk about is polymerase chain reaction, or PCR. This is a technique used to make lots of copies of DNA in a very quick manner. And it's used to copy a very particular region of DNA. This might be a case where a researcher needs to copy a genetic marker. It might be a criminal forensics person needs to match just a tiny amount of DNA that was found at a crime scene to a suspect. So the goal of PCR is to make enough copies of the target DNA so that it can be analyzed or used in some other ways. PCR is used in all kinds of areas, molecular biology research, all kinds of medical diagnostics, and even sometimes in ecology. Like regular DNA replication, PCR requires a DNA polymerase enzyme to make the new pieces of DNA, and it uses the existing strands as a template. What made PCR possible is a DNA polymerase that was isolated from bacteria in hot springs called Thermus aquaticus. The bacteria was called Thermus aquaticus, and its DNA polymerase is known as Taq polymerase. That Taq polymerase is very resistant to heat, and you'll see that heat is ultimately very important in the process of PCR. So Taq polymerase works best around 70 degrees Celsius, whereas human DNA polymerase works best about 37 degrees Celsius. So the heat stability of that enzyme makes it ideal for PCR. Like all DNA polymerases, Taq polymerase has to start building a new strand of DNA with a primer. This is a short sequence of DNA nucleotides that help it get started. In PCR, an experiment has to determine the region of DNA that they need to copy. Sometimes we're going to call that amplification, and they're going to have to choose primers that will bind to either side of that specific strand of DNA. So again, the PCR primers are short pieces of single-stranded DNA. Usually they're about 20 nucleotides in length. A PCR reaction requires 2 of those. And they're again designed to flank the target region, the region we want to copy. When the primers are bound to the template, they can be extended in both directions, and the region that lies in between them is copied. So here we see in blue the region of DNA, the template DNA, and then in the black brackets, it's the piece that we need to copy, the piece we need to amplify. So notice the 2 primers. They flank each end of the region to be copied. The primers bind. Taq polymerase extends the primers. The process of PCR really occurs in about 3 main steps. First of all, denaturation. That happens at about 96 degrees Celsius. This is when the reaction is heated up strongly, and this separates the 2 DNA strands from the original source. And this provides the single-stranded DNA templates for the next step. Step 2 is annealing. This happens at about 55 to 65 degrees C. So the reactions cool down a little bit so that the primers bind to their complementary sequences on the original source DNA. And then we get extension that happens at about 72 degrees C. So the temperature is raised up to about 72 degrees C. The Taq polymerase becomes active. It extends the primers and synthesizes the new strands of DNA. This cycle of 3 steps is repeated about 25 to 35 times. Usually takes 2 to 4 hours. And if it works the way it's supposed to, billions of copies of the target region can be made within those 2 to 4 hours. Here we can see the basic setup. Again, we've got denaturation, separate the double-stranded DNA source into single-stranded pieces. We get annealing where the primers attach, and then we get primer extension. What makes this copy so fast is that after every round, the new pieces that were created in that round can serve as templates for the next round. So essentially, it's an exponential growth kind of process, so that one or a few original pieces of DNA can be copied into millions or maybe even billions after 2 to 4 hours. All right, another topic we want to briefly talk about is gel electrophoresis. So this is a technique used to separate either DNA fragments, RNA fragments, or protein fragments, or whole proteins maybe, based on their size and charge. It involves running a current through a gel that contains the molecules of interest. And based on their size and charge, these molecules travel through the gel, the pores in the gel, in different directions or maybe at different speeds, and this allows them to separate from one another. Since all DNA molecules have the same amount of charge per unit of mass, in the gel electrophoresis process of DNA, the fragments separate based solely on size. So we can see, using electrophoresis, we can see how many different DNA fragments are present in a sample, and we can also determine how large they are relative to one another. We can even determine the absolute size of a piece of DNA if we incorporate into our gel what's called a DNA ladder or yardstick. And this is a set of DNA fragments of known sizes. So as the name gel electrophoresis indicates, we use this piece of gel-like, a slab of almost jello-like material called agarose. This gel has tiny pores in it that will allow the DNA or the proteins or the RNA in that matter to move through. At one end of the gel are pocket-like indentions called wells. This is where we're going to initially load the DNA samples into the gel. You can see in my picture the wells up at the top of each gel, and you can see that in the leftmost picture, a DNA sample is being loaded into those wells. You can see that once the power is turned on, and by the way, the wells should always be placed near the negative end of our electrophoresis chamber. We'll talk more about that in a second. Once the power is turned on, the DNA fragments are repelled from the negative end toward the positive end, and they separate into bands. And those bands are different size fragments of DNA. The smallest fragments are going to be able to move the fastest. The largest ones are going to remain closer to the wells. So before we put the DNA samples into the gels are put into a gel box, or what's sometimes called an electrophoresis chamber. One end of the box is attached to a positive electrode, the other a negative electrode. Where we put the gel, the main body of the box, we're going to fill that with a buffer solution that contains salts and that will conduct electricity between the two ends. As I said before, the end of the gels with the wells is always positioned toward the negative electrode. And here's what a gel box might look like. So we see our agarose gel in the middle with our sample wells. We've got a negative electrode on the leftmost side, positive electrode on the topmost side. That's hooked up to a power supply. And the whole box is going to be filled with a buffer solution that will conduct the electricity between the two electrodes. So we actually load the DNA samples into the wells, usually using a micropipette. And again, one of the wells is reserved for the ladder, and this is a standard reference that contains fragments of known lengths. We turn on the power, the current flows through the gel, and the fragments move away from the negative electrode because DNA itself is negative. The phosphate group within the DNA molecule, highly negative, so the negative electrode repels the negative phosphates. The fragments get pushed toward the positive side or the positive electrode. And due to the size of the pores, the large fragments move a little more slowly than the small ones do. So as the gel is running, the fragments separate based on size. And we get these bands, these well-defined lines called bands. Each band is actually a large number of fragments of the same size. We wouldn't be able to see a single fragment. So we have lots of fragments of the same size in each band. So by comparing the bands in a sample to the ladder, we can determine their approximate sizes. So if we look at this diagram and we look at row A, just to the left of row A, we have the ladder DNA. So if we compare the bands in row A, column A, to the bands in the latter column, we can see that the topmost band in row A is somewhere around 1,000 base pairs in size. And then the bottom band in column A is somewhere around 500 base pairs. If we looked at, say, column E, for example, so E has a fragment that's maybe just a little bit less than 1,000 base pairs. And then the small fragment is somewhere between 200 and 100 base pairs of size. Got a couple other quick points we want to make. We'll briefly mention the Human Genome Project. So the Human Genome Project was this international research effort to determine the nucleotide sequences of the entire human genome and to identify the genes that it contains. The project was run by the NIH, the National Institutes of Health, and the Department of Energy. It started really in about 1990 and finished up in about 2003. Its goal was to provide researchers with the tools to understand the genetic factors in human disease, and hopefully eventually opening up the pathway for gene therapies for those diseases, not only to treat the diseases, but also to diagnose them and maybe even prevent them. So because of the Human Genome Project, researchers today can find the genes suspected of causing inherited diseases in days, where before the project it might have taken them years to do that. There are now over 2,000 genetic tests for human conditions, and these tests allow the researchers and doctors to diagnose diseases very quickly. The outcome of this project is now leading to the development of treatments. And those are going to take time, but there are at least 350 of those now in clinical trials. All of the data generated by the Human Genome Project were made freely available on the internet so that all scientists around the world, not just those in the US, could use the information and develop these new therapies and treatments and diagnostic tools. And then the final thing we'll mention is pretty new. This is gene editing using a process called CRISPR-Cas9. CRISPR-Cas9 stands for clustered, regularly interspaced, short palindromic repeats associated with protein 9. Originally, this was a system found in bacteria evolved in their immune defense. Bacteria used it to cut up the DNA of invading bacterial viruses, sort of like restriction enzymes, that might have otherwise killed them. But scientists have been able to adapt the bacteria's molecular machinery to change the nucleotide sequences of an organism's DNA code. It might be the case that we want to correct a disease-causing error that was inherited or that happened due to a mutation. Or in some cases, we might be able to use CRISPR-Cas9 to enhance the genetic code of a crop, livestock, or even people. This might allow to the creation of like designer babies. So even though the technology is pretty new, pretty experimental, this is going to quickly lead to ways in which we can change the DNA code of not only plants and animals, but also humans. It's going to lead to lots of ethical dilemmas and lots of gray areas, but it could also lead to some really great advances in medicine and also in agriculture. So this is our presentation on biotechnology. Hope this helps.